My name is Steve Mayo. I'm the Innovation Specialist for TRISH at Caltech. One of the objectives was to bring in technology from across the country, and in particular in my case, to have a representative on the West Coast that could help source innovation, but also provide direct contact to researchers that otherwise might not interact with NASA. One of the other things I'm doing at Caltech is running a seed grant program that we call the Caltech Space Health Innovation Fund. And the objective with that seed grant program is to try to find investigators who have interesting technologies that could impact human spaceflight. One of the seed grants that we're funding this year is a project by Dr. Niles Pierce. Dr. Pierce has a technology that allows for computing at a molecular level the environment of a cell and then doing something that will respond to some input signals. One of the challenges with any gene therapy technology is getting the genes to express in the cells of interest, as opposed to having those genes expressed throughout the body. So the technology that Dr. Pierce has is incredibly relevant for that problem. He's been able to develop a concept called conditional RNA, where you can have an RNA molecule that will sense the environment, for example, by sensing a gene that is upregulated when you have radiation exposure. And then once that signal is detected, the RNA molecule can then trigger some other event. We have a project from Dr. Dawn Bowles at Duke University, and she is interested in developing gene therapy countermeasures for space radiation. And it's an interesting project because it fits into the cellular molecular category, because the objective is to develop molecular technologies as a countermeasure for radiation exposure. One of the risks for long-duration human spaceflight, including missions to Mars, is the elevated radiation exposure, and we're very interested in developing countermeasures for that risk that include gene therapy technologies that can be leveraged to target genes that are either upregulated or downregulated in response to radiation exposure.